Imagine comfortably sitting in a fancy clinic browsing through a local newspaper. Your partner brings your favorite Starbucks drink and a folder from the doctor. Inside the folder, there's an embryo catalog, where each embryo has a list of descriptions like gender, female, life expectancy 88 years, lower risk of developing autism, and a 60% higher chance of getting into an Ivy League school. This is what it would be like to design your baby. Here's the kicker. With the advancement in current genetic engineering technology, something like this might be possible. All living things are made up of cells. Cells are the most basic building blocks of life. They contain an instruction manual called DNA. Within DNA, there are specific instructions in a language called genes. Genes direct development, growth, and reproduction too. Sometimes, though, there are typos or variations in the instruction manual that can cause diseases like cancer. Now, what if DNA could be altered to correct these instructions? The answer lies in gene editing. We're going to look at two types of gene editing: germline gene therapy and somatic gene therapy. Germline gene therapy transfers a section of DNA to the reproductive cells, which are the sperm and egg cells. So the effects of this therapy are passed down to a patient's child and to successive generations. Somatic gene therapy changes the DNA of the cells that cannot contribute to reproduction. This way, the effects are not passed down to a patient's child. Currently, scientists have developed some gene editing technologies. The most recent is called CRISPR-Cas9. This is a revolutionary technology because it is more precise and more affordable for scientists. There are two key players in the CRISPR-Cas9 system. One is the Cas9, which is an enzyme that snips through DNA like a pair of scissors. The other is an RNA molecule that directs the scissors to a specific gene to make the cut. Once the targeted gene has been cut, scientists can then insert a healthy gene in its place. With such an exciting field like gene therapy, many myths and false claims are created. Here are a few myths about CRISPR and designer babies. Number 1. CRISPR can build customized babies. Scientists still don't know what every single gene in the body does or how it becomes what we see on the outside, because genes go through a complex process to be translated into physical characteristics. Factors like environment and little switches on the gene called epigenetics affect this translation process, and we still don't know how big their influence is. So because of our lack of information, we don't have the means to use gene editing to create a perfect baby just yet. Number 2. Gene editing will be available for widespread use soon. Gene therapy could be a life-saving technology for patients, but it's really expensive, and pharmaceutical industries don't like that. With these high costs, it's not likely that we'll have this technology perfected anytime soon. Number 3. Current genome editing technologies are perfect. Gene therapy is unpredictable. For some disorders, gene therapy will only work if we can deliver a healthy gene to a lot of cells and in its very specific location. Not only that, once the gene reaches its destination, it has to be activated and then able to function properly. Otherwise, it can cause even more harmful variations that cannot be reversed. If this variation was introduced using germline gene therapy, it can last for generations. The concept of changing a person's DNA, even to cure genetic diseases, is frightening to some people. Here are just a few ethical concerns to keep in mind. Are we playing God? For many cultures and traditions, gene therapy is viewed as playing God because it changes the fundamental aspects of the human makeup. Many argue that we have no right to do this, as scientists are tampering with nature. Equity of access. Current gene therapy treatments to create designer babies are expensive and very limited. Costs for one-time treatments range from $100,000 to $1 million, which means that gene therapy is just not a feasible option for most and would only be available for the upper class. This could cause division and may lead to further class discrimination in our society. Lastly, individuals produced through germline gene therapy can't give their consent for their genetic material to be modified. To what extent do we have the right to tamper with the genetics of another person, even if it's our own child? Gene therapy is something new and is continually developing as our technology gets better. Research needs to continue so that we can measure the long-term safety of these procedures. Using gene therapy to enhance or choose personal characteristics treads on the realm of science fiction. Yet, with most traits being the product of both genes and environment, we just don't know how altering genes will turn out. When we get closer to perfecting gene therapy, the chances of abusing this technology will increase, so it is essential that we have a system in place to regulate it. Lastly, we want to finish off with a quote. The traffic light for gene editing remains yellow and flashing. Proceed only with due caution. Thanks for watching!